Welcome to today's episode of Demystifying Science, where we have with us today Dr. Ted Pavlic from Arizona State University, who studies complexity. And this is a topic that is particularly close to my heart because when I was in graduate school, I was studying basically bacterial metabolism. And I became very, very disheartened by the idea that traditional approaches could actually allow us to understand much about metabolism as an entire system. Because you look at the map of what a metabolic pathway looks like inside of a bacterium, and you have thousands upon thousands of inputs and outputs and readouts, and many things are layering on top of each other to affect the same sort of readout that you'll get from whatever assay that you run. But it seems like you have developed the ability to look at very, very complex, perhaps even quasi-civilizational level systems and get something informative and valuable out of them.
Yeah, well, and I definitely appreciate that. Somehow when you look down at the complexity of these sort of systems, whether they be these metabolic pathways, like you mentioned, or at a level that is more close to sort of where I work, the level of say ant colonies, then it does seem like there's a lot of things that might be important, but it's not clear that they're important. And as you start studying them, it almost is not satisfying because you almost forget why you started to study that system in the first place. And so, like, if I was to look at a flock of birds, for example, I might be intrigued by the shapes that these giant envelopes of these birds take on. But if I really want to understand what's going on there, then I naturally want to track an individual bird and what that bird is doing. And as I start getting down into the micrograin details of it, I start kind of forgetting the forest by studying the tree. And it's natural to kind of zap all enthusiasm out of it because it just seems like this, how am I going to get from this individual out to this level here? So we've been trying to find ways in which we can kind of think about the macro as something that is just as tangible as the micro. And so one of the things like we've been doing, for example, is I work with this collaborator, Jürgen Liebig, who studies these beautiful Indian jumping ants. They're called Harpagnathus saltator, Jordan's jumping ant. They're very charismatic. So they're these ants from India that are about, say an inch long or so. And they have these giant mandibles that are sort of actually forward. And they're that way because they can hunt for live prey. And so they jump and they can pierce this live prey and carry it into their nest. And there's all sorts of interesting things about their life history that are distinct from other ants. And one of the kind of important things about it is that workers, so female workers inside the ant colony, in most ants that we talk about in sort of Ants 101, are going to be effectively sterile individuals, sort of an extended phenotype of the queen. They're just kind of there as sort of cells that have flaked off from the queen and are able to sort of do the queens to help raise the other offspring. Now, these ants have retained the ability to mate. And they can not only mate with, in general, but they can mate with their brothers and even their sons with no inbreeding depression. And so after a colony has lost her queen, then the workers can themselves start mating and or have stored up sperm from previous matings and take over. And so what's beautiful about this process is it sets up kind of this example where we can watch a social transition at a time scale that we normally can't watch it at. And so there's this famous experiment from Australia about watching the pitch drop, where they take tar and they pour it into this kind of funnel, and then it starts forming a drop. And as that drop forms, it takes like 10 years to actually complete that process and actually pinch off and fall. And so you can record this thing in high-def video and really understand the fluid dynamics of how this happens. And we realized this was a system where we could sort of understand at a high, fine-grained time scale how social transitions happen. So we can remove the queens from these Harpagnathus, or a grad student named Ben Pineson can remove the queens from these, start them off in this sort of disoriented state, where then they go through a 21-day roughly process of electing the next committee of egg layers. And it's important to pick just a few of them, because whoever gets transitioned into this role actually stops being able to do that hunting that we talked about before. Their brains actually change, their ovaries change, and they just totally become a completely different animal. And eventually, once you get a couple of them, then the process stops, and the colony goes on until all the reproductives are removed again. Now, in the past decade of people, say, studying these ants, or a little bit longer than that, people have realized during that 21-day period, there are certain, like you say, certain metabolic pathways, you could call them that seem to be important. There's, for one thing, you can see ants will periodically do this dueling behavior where they go up nose to nose with another ant, and they go back and forth and back and forth. And they aren't actually attacking each other. It appears to be triggering internal changes that trigger this kind of physiological difference. So it's necessary, but it's one of the things that's extremely conspicuous. But we're wondering, are there other things that could actually be going on during this period that could help give us a better idea of how you get from one macro scale state of sort of no hierarchy to another one of hierarchy? And so what a student of mine did is Taeyoung Che. He said, well, what most people would do is track every single ant individually, like those birds, or tracking every single metabolic pathway. We realized that simply putting coordinates on every ant. So this ant is in this position, this ant's in this position, in the next frame, they're in slightly different positions in the next frame. That was extremely tedious to do. And then you also then lose a bunch of information because you've gone from this picture of this big, beautiful ant that has all of these different behaviors. It twists its body in different ways. It's very informative watching these individuals, turning them just into coordinates. We said that is one way to go, but there's probably other ways we could do it. So what Taeyoung figured out how he could do is you can take these 21 days worth of video and pipe them into a deep neural network that he specially designed so that it can look at the raw video data, and it can be trained to say, these are video segments, little 30-second segments from these 21-day videos that come from particular times in the hierarchy. They're at day 1, day 21, day 15, and so on. And what he was able to find is that without giving the neural network any information about what we know about the ants, about dueling or anything else like that, it was able to take 30-second video segments, ones that it hasn't been trained on, new ones, and predict pretty accurately where they fall in this 21-day period.
And is the idea you're looking for actual events that constitute the decision-making process in this group behavior shift? You're kind of teasing out the decision-making back to the...
Right. So, I mean, the problem, so what motivated this whole thing is that before we did this, our collaborator, Jürgen Liebig, would have a grad student just sit and watch video for huge amounts of time looking for interesting things that were going on, interesting from a human perspective. But it's hard for, you can't like watch the whole video and sort of go back and kind of take it's different. This is not a very good human task to do, to sort of screen these videos for possible behaviors. And so our first question was, could a neural network possibly be able to, given a small segment of video, predict where it came from? In other words, could it key in on certain things that were happening that kind of showed we were later in the hierarchy formation process or earlier? And that's the first thing he did, is create that. Then the second thing he did was build a second network that could look inside the first network and ask it, okay, there's got all these complicated, ugly, latent variables inside the brain of this first neural network, and was able to say, can we look at those and say, for any given video segment, which parts of the brain of this neural network are lighting up? And can we visualize those back on the original video, so that he could then put little spotlights on areas of the video that appear to be informative in order to make the right decision.
And that's where these novel behaviors are occurring at? You're hoping to discover these decision-making features?
Right. That's right. And so what he found on the first pass here is that it discovered things like dueling. So it was able to highlight the dueling that we knew ahead of time was important, but we never told the neural network. So it was able to find that sort of thing there. And so now what we've done is we've gone from a huge amount of video data to now a bunch of these sort of slices, frames from these videos and with highlights, where the neural network has said, here's something that was helpful to me. I don't know, this might be totally by random chance, but I think you should go and investigate this.
Mm.
And so now, instead of having a grad student watch all 21 days of these videos, we can now have even people who are less specialized knowledge look down through these and say, look at all these kind of pre-screened, slight frames from these videos. Do you notice commonalities? And from there, we can form new ideas about what might be going on. But then we could go.
So, what are some of the new ideas that have popped out? Have there been new
behavior? Before we move on to that, I actually really want to ask about the way that the neural networks are trained. So you say that you train it, but you don't give it any information about dueling or the behaviors that you know. So what does it actually entail to give a computer program a video and then consider it to be trained afterwards?
Right. So we can feed it. So this is a sort of kind of a supervised setup here where we can feed it examples of video segments that come early in the process and later. And we can tell it that this is a day 1 video, this is a day 21 video. And we can give them that from different colonies, for example. And then each video, there's all sorts of these like little tiny data science technical things. Like you want to make sure that it's looking at the ants and not looking at the plaster that the ants are walking on. And so we have to do certain things like, well, maybe we don't give them the raw video exactly. Maybe we can give them what we call like an optical flow. So we give them actually sort of the rate of change from one frame to another. Or maybe we rotate the video several different times. And so it's not just trained in one orientation, it's trained in multiple orientations. There are all sorts of things we have to do. But the essence of it all is we can give it a video, we tell it what day the video is from, and here's the video segment. What day the other video is from, here's the video segment. And we have a whole training set of those. And then we have a test set of video segments that we know the day, but it doesn't know the day. And then we can say, how well did you predict the day? And it turns out that it can do pretty well.
And you're comparing it on videos of roughly the same group of ants, or you're using different group of ants. So if you train it on, so it seems to me that the way that you create these disordered conditions is you take away the hierarchy by removing the queen. Then there's this dissolution into dueling and behaviors and committee making. It takes 21 days. On the other side of it, you have a queen. Do you then, after training with videos of that process, do you repeat the process with the same group of ants? Or do you have a different colony where you remove that queen and look at that? Like, how translatable is it across?
Great. Yeah. Well, so one of the great things about Harpagnathus is we can create multiple colonies of roughly the same size, same number of brood, and so on. And so in this particular study that I talked about here, which is you just had published, is that we used several colonies of the same size. Now, since then, while we were going through writing that up, Ben, the animal behavior student who's attached to Jürgen's lab, has gone through and created a database of a wide range of videos from other sizes. And so we can then start to see how well do these things generalize. And we can start answering that question. So right now we know that given that we train it on roughly, say, and this particular species of ants lives in relatively small colonies. And so we can train it, let's say, on about 60 ants. And it appears to have generalizability across other colonies, which also happen to be around, say, 60 ants. So then the question is, does that generalizability hold for 20? Does it hold for 150? And if it doesn't, is that because there's a fundamental shift in how the societies work? Or does that just mean that it's become more cryptic because the density's increased and now the neural network doesn't know how to deal with density and we have to sort of figure out that? So that's kind of the next step. And so likewise, the earlier question about what new things have been found, we are currently mining through that data now. Right now, as a proof of principle, we were just excited to see that dueling came out and other behaviors that look dueling-like. So sometimes you can have 3 ants kind of come together with their heads pointed towards each other. If I just showed you that, then superficially you might say it looks like they're in a duel. But the neural network was able to actually tease out the differences between duels and things that aren't duels that we, at least as humans, would think look like duels. So then the next step is to go down through and all the other candidates it's generated and see, are there other things there? And we don't think this replaces tracking individual ants like the kind of standard ways things we're doing, but we do think that this general idea of training neural networks on, it really comes down to just control versus treatment. Like, we could, in principle, we could have other types of conditions, things that don't change over time. But you might say, here's a bunch of ants that have been treated with a particular drug. Here's a bunch of ants that are low in oxygen. Here's a bunch of ants that, you can imagine a bunch of different treatments relative to some control. And not knowing exactly what behavioral outcomes could come out of that, we could use a similar system to say, there's a bunch of anti-like behavior that we can factor out. And we could see that actually, and now people already do this, but they do it with very coarse grain metrics like the density increased, the average speed increased. With this, we hope to actually be able to say, look, there's actually more of this weird behavior that occurs where two ants crawl on top of each other or something like that. And that only really occurs in the treatment group. And that's something that might be really easily missed by a human observer who's kind of zoomed out and just looking for like how frenetic the ants are.
So basically, you're fighting against this tendency to reduce the information to locations. Do you imagine, can I zoom out from this and kind of ask you to consider what other applications you could imagine this sort of training pattern to be efficacious in? Like, it seems to be perfect for discovering new targets for molecular disorders in the body, things like that. Is there anyone working on that to your knowledge, or what are some of the other applications that you've
do you have an answer for metabolism?
Yeah, so that's a little bit outside of my area. I can tell you, I went to when I was in grad school, my office mate was doing computer vision work with swarms of bees, and he was trying to figure out which bees. So back in that time, there were two hypotheses, and so there was blobs of bees would fly from one point to another, and these and only say 20 of them would actually know where they were going. So this is after they found a new nest, and so at that time there was a hypothesis that you either could have so-called gentle guides, where there are these bees flying roughly at the same speed of other bees, but they had a little bit of a bias in themselves. And so because they tended to want to go in one direction and they were sort of stubborn, then the rest of the group would kind of go with them. And so people like Ian Cousin and so on who study fish, they were kind of advocates for this gentle guide hypothesis. But Tom Seeley, who was my colleagues, my office mate's collaborator, who's a really big bee guy up in Cornell, his anecdotal experience was that there were always some bees that were flying really, really fast through the swarm, much faster than the rest. And it was his thought that it's possible they could be actively steering. And I swear I'll get back to your question in just a second. But Kevin, my office mate, then was able to generate sort of some computer vision that was able to track all of the bees from like looking upwards at them, and was able to find that there was this small subgroup of bees, and they did seem to be kind of steering the colony in the way that kind of you'd expect from this sort of what they call a streaker hypothesis instead of a gentle guide hypothesis. Now, Kevin used the exact same stuff.
They're like bee dogs, like bee sheepdogs.
That's right. Yeah, they were kind of corralling or herding the bees, yeah, in that sort of way, right? But Kevin was able to, along with that, also took data sets on these pancreatic cells that were kind of these these blobs that were moving around, and he would try to apply the same sort of thing to see can you predict the motion of other blobs from the motion of certain, are there ones driving the kind of collective motion and so on. And that ended up being kind of to be determined because it turns out the bees were much, much easier to track because they have so much more inertia. They're a tiny little nut that's flying through the air that doesn't turn on a dime. And all of those things end up being much more helpful. But still, the insight was there. So I guess what I'm trying to say, there are people who are definitely looking at those. I have a colleague here at ASU who's looking at the movement of cancer cells in the brain, and because they have a thought that actually they're foraging for oxygen, and so is the oxygen availability and actually cause them to sort of stick to certain vascular structures, and sort of trying to understand is there a highway in the brain that the cancer cells have to use. So there are definitely people who are looking at these questions. I don't look at these questions specifically. I definitely think we could apply that if we had the right data set. the broader impacts we've kind of considered have been more at even the sort of a human social level, where once we've looked at this sort of pitch drop, we can understand these social transitions over long periods of time. If we learn kind of important generalizable cues, then we could say, well, let's look at video in a mall or something like that. And are there things, things that are relatively simple? We wouldn't need to necessarily send them up to a cloud for analysis, so for privacy preservation, that might indicate that, there's something that's off nominal about this behavior that might suggest that maybe a riot is about to break out or something along those lines, and that might raise red flags and say, like, well, maybe we need a human to look at this. So it might sort of help reduce the cognitive load on human security guards, because it would help prioritize, which videos to look at. And
it's future crime detection. It's like, reminds me of a Philip K. Dick book or something.
Minority Report, I think, yeah.
Minority Report,
right.
There you go.
Well, I mean, even years ago, there was an article in Control and Decision, or IEEE magazine on control systems, Control Systems magazine, that actually was using drift diffusion models from cognitive science to characterize how good a human was at making a decision about a video in front of them. And at the point at which it seemed like the human was going to be effectively making a random choice, like, you've looked so long at this video, our models suggest any decision you make is going to be random, it would switch to a new video and say, let's look at something else. And so, that's kind of the things that we're sort of thinking about, is that we're not necessarily trying to have predict what's going to happen, and have the computer be the arbiter of that. But we're trying to come up with these augmented intelligence schemes that consider the dynamics of the system you're looking at, and also the dynamics of the cognition inside the person, so that you can make them a true team, where it's really knowledge co-creation and actuation, so that we're not asking one to do everything, if that makes sense.
You've used the term intelligence and brain and vision in relation to computers and computer programs. And these are terms that I feel get mentioned without there being a universal agreement upon what it is that they're implying.
Mm-hmm.
So for me, when you say that something has a brain, I immediately imagine an organism or a system that is not only capable of learning, but it's capable of innovating, it is capable of coming up with novel solutions to things. But oftentimes when I talk to people who work on computer programs, it comes down to the fact that there's a training set for a specific stimulus, and then the computer can work within that stimulus, but it's never going to suddenly develop an ability to look for a new stimulus, unless you specifically train it to do so. Is that the programs that you're using are, when you said that they have a brain, when you say that they can see, what does that actually mean?
Right, that's a great question, and also, the question of, like, what is a brain? So I mean, I walk in a couple of different disciplinary circles, some with physiologists, some with behavioral ecologists, some with engineers, and depending on which group you're in, the significance of having a brain varies. And so you to some of the people that I work with, a brain really has to mean that decision making is centralized in an organism. So when is it useful for there to be a central center of decision making? And so whereas there of course are theories out there about animals, a lot of animals have brains and plants maybe don't, because animals have to move themselves around and plants are planted and animals are animated. And so you need a central area to coordinate the motion of all of these things. And that actually, fits with things like when we look at ants, there's a lot of ephemeral brainness in ants. And so I mentioned bees, for example, they're highly related to ants. That there's, when they move from one nest to another, only 20 bees out of thousands know where the new nest is. And so and those 20, once they hit the new nest, fold back into the rest of them and stop being the kind of the migration bees. So it's like, when temporarily, when you needed to move everybody, then there was suddenly a group that took control moved everybody, and then disintegrated back into the background again.
So the brain is like the embodiment of the decision-making process?
I would say that yeah, like sometimes we say, what is a brain? And I would say a brain, it would be a centralized location for information processing, where most of the important information gets to, and most of the important decisions are made and then actuated from. And then, but then so that's the reason why I love looking at bio. So as an engineer, I love looking at biology for looking at all sorts of exceptions and diversity. So not every animal even has a brain. There are some that have nerve nets, for example, that are a little bit more distributed and things, and yet they still manage to do. So that's it comes back to the question of when do we need a brain? And if I were to translate that engineering, then I can say when do we need so right now there's a lot of work in engineering and computer science on so-called federated learning. So there was a big push to put everything up into the cloud. So for the longest time, when I was in grad school and engineering, the people would say, how are you going to have the computational power to do that? Like somebody would present their dissertation and it would show some really interesting results on autonomous cars or whatever. And then there inevitably be somebody would raise their hand and say, how long did it take you to generate that trajectory? And they'd say, oh, on my super powerful desktop machine, it took a week or something. And that was a trajectory for one drive, that was supposed to happen in the morning of, some car or whatever. And they'd say, well, how do you expect that to scale? Like, and then people would always then say, well, it's in the cloud. So eventually you'll just be able to put it in the cloud and it'll happen instantaneously.
Sufficiently advanced technology is indistinguishable from magic.
That's right. Exactly. Right. Yes. You're counting on magic. And for a long time, that's where everything went. And then people start realizing that although we could make magic in the cloud to some extent, maybe we don't want to be giving the magician all of our biometrics or all of our details and things like that. And so now federated learning was sort of this idea that, okay, we're going to take everything away from the king and kind of concentrate power in the states. We're going to create kind of this federated thing where out on the edge is where we're going to have the computing. And so we're going to do a lot of information processing on the edge and then send anonymized things as needed back to a central area, and then get anonymized stuff back from it. And so that way we could somehow still gather information from across the globe, but we would still keep most of the fine grained details localized. And so once you get into these sorts of architectures, then it is hard to say, well, where is the brain in that machine? The brain seems like an abstract idea that is implemented on a substrate that is spread all over the place. And so
I think that paradigm's kind of shifting in biology too, though. The more that you look at, especially the involvement of the gut and goes, the gut feeling, and honestly, there are all sorts of immune decisions being made all throughout your body. So
absolutely,
it's interesting how that idea of the brain is kind of dissolving, especially the more we understand the immune system and the way that it interacts with the nervous system.
Right, and I think that's a great point, and that's why. So for me, like I said, I travel in a bunch of circles, but I mean, the one area of biology that's close to my heart is behavioral ecology. And in behavioral ecology, there is a fundamental distinction between mechanism and function, and on the side of that also between mechanism, phylogeny, ontogeny, development, and function, and so sometimes when I have to ask somebody, what do you mean by brain? I have to ask them effectively, are you referring to brain as function or brain as mechanism? So there's that wet hunk of matter that's underneath my skull, and you can ask questions about it, but those are mechanistic questions. Or like you just said, effectively you can ask functional questions about information processing, and we can refer to that as being a brain colloquially, but really we're talking about something function. I know in neuroscience, there are people that are now debating this, like for a long time. So cognitive sciences have sort of asserted that there are modules in the brain that do not necessarily relate to true, to true structures that actually exist in it. Like this is an attentional area. There must be a module for emotion, these sorts of things. And we just sort of take them for granted that these things actually exist. And I think to a behavioral ecologist, they would say, well, it seems like you're mixing function and mechanism. And I'm now starting to see conversations in neuroscience where they're actually calling for people to to be more formal about these things and say, don't refer to an attention center of the brain, refer to attention as a function, and then tell me about how different mechanisms in the brain contribute to that function. But we can study those as separate entities. And so I guess I would apologize for using the word brain earlier. I guess it was just convenient. But I think it's a very deep rabbit hole to really say, when we talk about the brain in conversation, what are we really getting at? And that comes back to my last point there. Are we getting at a function, or are we getting at a mechanism? And once we're clear about that, then maybe we can have more intelligent conversations.
I love that. Yeah, it's a huge problem too. I think at the cutting edge of physics, confusing material with effects, especially things that we understand and harness so well in engineering, because oftentimes the functional description, the quantitative relationships are sufficient to be able to develop pretty powerful tech without actually concerning yourself with the material actors involved. And so it's cool to hear it laid out that way.
Although, it's interesting you bring up engineering. I mean, so I teach a lot of engineers about how to create intelligent things and all that, and how to compare to biology. And it's interesting to me that the conflation of function and mechanism, I feel like, is still very strong in engineering, even though engineers have to build things, because they will assume that the things they built to do the thing they're going to do always do that thing. So you assume, like, you could say, well, how do I make this robot find a light source? And I'd say, well, follow a photo gradient or whatever. And so, and so they build some mechanism that causes the robot, hopefully, to move toward a light source. And they would just assume that like, they might even implement some form of gradient calculator inside inside the brain, inside the central processing of the robot. And just because it's calculating a gradient doesn't mean that gradient is actually the physical gradient.
It's just the output from whatever your photo resistors are telling you, or whatever.
Right. Or even if they've got some sophisticated mathematical model that filtering the photo resistors through, like the little CDS cells go into a model which estimates the actual gradient, and then they get a numerical value of the gradient, and they try to climb that gradient, and they'll tell me, no, it's climbing a gradient. I programmed it to climb a gradient because it estimates a gradient and climbs that thing. And say, well, most of the time you'll be in an environment where the gradient inside the head of that robot will be the gradient in the physical space, but that need not be the case.
Mm-hmm.
Like, you just happen to build a mechanism that's pretty darn good at its function, but I bet I could put it into an environment where we would get really confused. And so we have to be clear that just because you wrote gradient climbing into your code doesn't mean it's actually going to climb a gradient.
And what are the conditions under which you would take a robot that has been programmed to climb a gradient and confuse it sufficiently for it to not do that?
I mean, so you could maybe in a mirrored area or something like that. I've got colleagues who, I've got a colleague who he does a lot of work with computer vision in autonomy, specifically big monolithic robots and cars, and he really believes that it's important you can't just train a neural network to do computer vision, that you really need a knowledge backend as well, and those two things can kind of work together. And that's in order to make sense of weird things in the world. And he brings up these points to, like, if you took most autonomous vehicles, that are driving, in test, like, I live near Tempe, Arizona, and Google drives a lot of its own cars around, or Alphabet, Waymo drives a lot of its cars around, and you see these cars driving around working fine. Imagine if they pulled up behind a car that, and this is an example I borrowed from him, that just had a giant mirror on the back of that car. That would be weird to me if I pulled up to it, but I would suddenly realize that I was looking at me and it was looking at a mirror, and I could deal with that. The autonomous vehicle, now granted, it has more than just vision sensors, but if you just focus on the vision, that's going to totally upset the vision system. So that's kind of an example where you really, like, the mechanisms that work under a non-mirrored world are going to be totally broken under a mirrored world, and yet mechanisms in my brain seem totally fine with that.
And you can imagine all sorts of interference messing with the initial input receptors in your robots or whatever else too, that could look like it was being overloaded with voltage when it wasn't. Or ultimately, it's only capable of reporting if its function is What's functioning? What's functionally happening? What's
Well,
happening?
and on the flip side of this, and this is something else that we study and that we're really excited about, is that a lot of times at the micro scale, when we see little errors in sensing and sensory motor actuation and all that sort of stuff, we initially, it's tempting to call those errors. And if we were engineering the systems and we saw these things, we would say, well, we need to get rid of that. We need to shield this cable, or we need to put in these filters or something like that. But in the biological systems, those errors are often sort of embedded as important parts of the system. So as an example, there's if you look across, a lot of people know about ants and trail laying. And so not every ant lays trails. So that's something not everybody knows. But ants 101, you hear about ants and trail laying. Now, certain ants, like Lasius niger, which is the garden ant that you see in Europe a lot, that ant, when you watch it lay trails, it follows those trails like perfectly. And so ants just kind of walk down those trails. It's a very good trail layer. And you can put out feeders for these ants. And they'll, if there's like a good feeder, like it's got a lot of food in it, a bad feeder, it doesn't have that much food in it, then it'll go to, they'll scrutinize the two feeders and eventually collapse to one, a line going to the good feeder. Now, what they realized back in studies in the 80s, early 90s, is if you take these feeders and you swap them, so that now the bad feeder's in the good feeder place, the ants will swap so that they'll now find the good feeder again. But then if you swap them back, sometimes they're not very good at sort of tracking the other way. So, or in other words, if you remove a feeder, they're good at resetting. But if you swap two feeders for each other, they're just not very good at tracking these things back and forth. And mathematical models, simplistic mathematical models of trail laying, kind of suggest that actually behave the same way. And so we just sort of assumed that these ants were, they were using a simple rule and it worked most of the time, but it would easily be tripped up. Now, cut to maybe 10 years later, and people started thinking that maybe noise and mistakes could be useful, and maybe ants could be smarter than Lasius niger. So some other people I know down in Australia who did this initially. There's other ants that are pretty good trail layers, but they're just noisier about their trail laying. And so they speculated that maybe you get a different result with these ants. And so they gave the ants a lot of these other ants, these Phaidole dentata, for example. So there's these other types of ants that happen to lay trails. But when they lay a trail, an ant will come up to the trail, and she might not always follow the trail. Maybe 20% of the time, she'll come up to an area that definitely has chemical on it and just go another way. And what was cool about these ants is when you put them in the same sort of assays, they have no trouble tracking the good food as you switch it back and forth. And when you look at what's going on, it actually looks like the ants that are making mistakes are constantly putting a population of ants out in areas that might sometime be a better feeder. And so the mistakes are really sort of a bet hedging strategy in order to deal with the fact that sometimes they live in ephemeral environments where sometimes you have to change things up.
These are the artists. These are the artist ants. They're out there on the edge exploring. Right. 9 out of 10 are terrible ideas, but every once in a while,
That's right. That's exactly. And that's one way to interpret it. Is it's like most of the time they're wasting all this effort, but in reality, because fitness is assessed at the level of the colony, really, we don't want to give a score to an ant. We want to give a score to the colony. That's where function lives, at the colony. Function doesn't live at the ant. We don't want to call an error an ant. The error is the Lasius colony that can't track. So I don't care how well an individual ant follows a chemical trail if it means their colony starves. But there are downsides to putting 20% of your foraging force out exploring where usually there's no food. And so not surprisingly, Lasius happened to evolve in an area where things were more steady, and Phaidole happened to evolve in areas where things are more ephemeral. And so some people have called this kind of a stochastic resonance in ants, where there's an optimal noise value that every ant has to make use of in order to properly sort of shape its recruitment patterns. And so, I don't know, the whole reason I'm bringing this up is like, we have to sort of ask, where are we assessing function? And when we start assessing function where it actually kind of rears its head, like at the colony level, things that we would have called ugly noise or mistakes or sensory limitations or whatever might themselves actually be a mechanism that really contributes to function.
And this is the same as the junk DNA in the 90s, where there was a gradual push towards realizing that, well, it might not be reasonable to think that 97% of what is contained within the genetic material isn't actually a useful aspect.
Right. That's a great point. And it's funny, and now that so there's this area of evolutionary computing called genetic programming. And in genetic programming, there's a bunch of cool things you can do with it. You basically can have a computer write code for you. So you can say the code has to do these 10 things, these unit tests or whatever. Like maybe it always has to count the number of letters in a string or whatever. But then you can also put pressures on it. And I want you to do it using as little energy as possible, and so on and so forth. And so you can, using as little code as possible, or as little memory as possible. And you might not know how to come up with a novel algorithm for this particular thing, but you just let the computer evolve. So it creates populations of programs that compete against each other. And then it promotes those that do well and demotes the ones that don't. And then at the end, you get a terminal population of programs that may have done something that is novel. And so one interesting thing that happens in these genetic programs is you get an accumulation of junk code. So like for anybody who's done kind of programming out there, you can imagine like if I had a loop that had said like while false, and then anything inside that loop would have like a bunch of code in there and then end while, that loop would never be executed. So it would be like an intron in DNA, So, but with one little change, like if that while false gets mutated into a while true or while some condition, suddenly that code gets turned on. And what they find is that if you prune away this stuff in the process because it's not operational, you tend to find that your programs don't evolve quite as quickly. So there's these little like miniature experiments going on in the junk that are kind of like, they're sort of walled off. They can't do any harm while they're behind that while false. And they can be, and they can then take evolutionary leaps forward in function. Whereas if you didn't have that, you would have to, every single minuscule change.
You'd actually have to build it from scratch.
Well, yeah, and every minor change would have to keep from breaking the function of the existing code.
Because nothing could step up, sort of. It's like shark teeth. You lose one and you have rows of 3 that can come forward.
Right. Yeah. And so you kind of need a sandbox on which you can play with things. And then periodically, unleash whatever you developed in the sandbox. And maybe that'll be terrible. But
that makes a really interesting argument for like a free and open not only society, but business plans for the way that you actually invest in your innovation as a civilization, because it seems like you need this baseline level of wealth. You need this reservoir in order to pay for folks to sit around and make mistakes sometimes. I don't know if you've thought about how this applies to like civilization level structures, but it seems...
in a more utopian way, because I keep thinking about the sort of dystopian version of security guards in malls watching programs, directing things, but this idea of using the same principles to build positive outcomes for development? Is that kind of where you're going?
It just seems like there's definitely a pressure in civilization for people to get on, a tried and true track with their lives. It's been really interesting just from a personal standpoint, being an artist and being a creator, but also having gone through the academy and worked in science and worked in these huge institutional machines. There's always this tug of war between spending time on creative processes. You see this divide between labs, the way that PIs handle their labs. I had an amazing advisor in graduate school who, when I showed up, essentially, I did some rotations and I chose this lab, and he said, "All right, go ahead and play for the next year. Like, just we knew what system we were going to work on, and he was just like, just look around, just see what you can find that's interesting here." And that's an insane thing that almost no other PI that I knew of did at the time. And I think it was really amazing because we did discover something really cool eventually. And it took a while. It took a lot of tinkering and building new detectors and trial and error and not really coming up with anything to say, not having anything to write a grant about. It's a risky decision to do that. And so I think what you're seeing this pattern play out evolutionarily in nature is something that could really be taken home to the business level, to the institutional level, in terms of just reinforcing the value on dedicated, wasteful, creative time. Like, it's not necessarily wasteful in the end, obviously.
It's just not directly productive, right? I think that we do have as a society, we have kind of decided that the things that we will value is the labor that will be immediately productive.
Mm-hmm.
And yeah, it does really seem to suggest that biological systems function best when there's a whole bunch of labor that might be walled off, that's not particularly productive, that at a moment's notice can be sort of pulled up to take the place of something that is crashed and burned. I think about this all the time in terms of retracted research, right? You have these huge laboratories that are publishing in big papers and big journals, and everything's very fancy and they're doing great. And then all of a sudden there's somebody who's working in the background who's like, I don't think that's how that works. And then the person at the top falls, and then the other person is able to step up and be like, look, this is the direction that we should go. But if you haven't published for a long time, if it's been years between successes, it can be very hard to maintain the funding from university or government levels to actually pursue that.
Right, and that's a great point. I do want to say, so as a caveat, the little genetic programming example, that is sort of suggesting that in biology that these introns are junk until they're turned on, which of course now we're finding out that they might actually be being used even if they're not. So there's probably a lot more interesting stuff going on in biology than the kind of the dumb programming example. But I take all those points, and I think your point there about sort of what we choose to. I mean, you could argue that in the private sector, things like venture capital really are expecting a very low rate on return, and they're constantly spinning up companies that go down. It's sort of unfortunate from the research side of things that we aren't learning that much about it. And I'm not going to try not to go up on the soapbox too much here, but it's state institutions, major universities. Once upon a time, or at least I know a lot of my colleagues got into the faculty route because they pictured a place of intellectual freedom.
Pipes, elbow patches.
That's right.
Velvet paneled libraries.
Right. Where you go in and you come up with crazy ideas in the morning and you play with them all week, and sometimes they don't work out and it's all fine.
Instead, you find yourself screaming at grad students at 3 a.m. after their experiments aren't working and things.
Well, yeah, and you ask, well, what are the pressures there that have caused that? And you could look at there's now once upon a time there was much more investment in the public in the public university, and so we once viewed the public university as a public good. It was an institution that we pay for that produces ideas, it produces talent, it is where people go to mature intellectually, and so on and so forth. But gradually that became less palatable for people to want to fund. And so I teach a lot of courses in sustainability. We talk about, what's the difference between public goods and private goods and so on and so forth. And really effectively what we've done is, even though we call public universities public goods, because they still are governed by state charters, they still get money from the states. And but if you look at their budgets, a huge amount of the support for public universities comes from tuition. And you might say, well, that's fine because those individuals are going to get an education. It's going to be valuable to them. They should pay for that education. And sure, but that changes the nature of the university to being a private good that somebody has to pay for to get out of it. And it also means that instead of the universities trying to generate public goods like ideas, the universities try to generate things that attract tuition dollars. And so there is going to be a pressure on a faculty member to become very prestigious, because if they become very prestigious, then that means a faculty member at another university is going to recognize them when US News and World Report sends them a survey. And that's going to then move that original university up in the rankings to get more students and parents who look at those rankings to enroll in that, and so on and so forth. And so the chase of tuition dollars largely changes the character of institutions, where it becomes much harder to be, I mean, and if you think about it too, like, there's other sort of trickle down effects like that, like TA availability, in courses. And so a lot of their students don't think about, they don't know what their TAs are, and they don't realize their TAs are grad students who are being paid to help participate in the teaching process and so on. But the grad students also have their own dissertations to write. And so we're subsidizing the generation of really cool ideas by giving people TAs. And as TA availability gets less and less and less, what we're basically saying is that grad students have to be funded either by fellowship on their own or by RAs alone, which is on targeted research projects. And it's going to be a lot harder for them to go off and investigate these other things. So again, I don't want to get up on a sort of a soapbox here, but I do think that it's interesting that the venture capitalists are still willing to take more risks than I think the public is when they invest in public universities.
Mm-hmm. I mean, to tie that back to the machine learning and analysis of how systems work or don't work, is this loop something that you could study and parameterize well enough to then be able to say an intervention at this point and this point would be the most effective at creating the desired outcome? Because it often seems like you talk to university professors, you talk to students, it seems every single stakeholder in this situation looks around and agrees that it's not in a very good state. So it seems like it would be relatively...
Or at least an antiquated state.
Or at least, right, it's not modern or updated. And so is it possible to create programs that analyze real human systems like this to be able to say that these are the pivot points, and if we do these 3 interventions that everyone can agree on, we'll get to where we want to go?
Well, I think so in principle. Now I do a lot more kind of armchair, fundamental research where, like, I'm studying ant colonies, right? So it's hard for me to extrapolate with a straight face to a lot of human systems. But there is some work that I did with an old colleague, Taka Sasaki, and a woman who works in homing pigeons, Dora Biro, really well known for homing pigeon research. And so we used, with the lead author in this work was a postdoc from my lab, Gabriele Valentini, and he used these frameworks called transfer entropy that sort of are able to, if you give them large spatiotemporal data sets where you can imagine each trace is a different individual's actions. And so I have your actions through time, my actions through time, someone else's actions through time, and so on. And I don't know who is interacting with who, but I can look at these things and I can say, do I notice that every single time person A does certain behaviors, person B does certain behaviors a little bit later? And if I start seeing these sorts of things, then I can start forming these type of, I don't want to say causal links, but they're meant to kind of be an indication of possible causality. So they're still correlative, but still that you can kind of get an idea of who's doing what. And so these sorts of things revealed, at least with these homing pigeons, Taka and Dora came up with this really cool, I don't know if you've heard of the spaghetti tower experiment, but it's sometimes you do it in like team building exercises, and you give a bunch of people just a box of spaghetti, and I forget what you put, like you give them maybe some bubble gum or something like that to like fasten the spaghetti together, and you'd have them to try to build the tallest tower. And then so in the spaghetti tower, there's this like cumulative cultural evolution experiment where like 2 people at a time keep building the tower. And so you have like 2 people work on it for a half hour, and then you swap one of them out and bring a new person in, and swap one out and bring a new person in. Or you just have 2 people work for the entire 4 hours on the spaghetti tower. And it turns out that by swapping the new ones in, you actually get much taller towers. So it's kind of an interesting thing. What Taka and Dora figured out is you can do the same thing with homing pigeons, where you displace them, miles away, and then you release 2, and you watch how they fly back to their home, and then you displace them again. And then you release another 2, where one came from the original group, and then another one was a naive bird. And you can do that over and over again. And if you do the same 2 over and over and over again, you can see how well does the route improve over time. But it turns out the route improves much better over time if you keep swapping in a new bird. And so what what Gabry did is he looked at traces from these and analyzed them with transfer entropy and noticed that there would be bouts of kind of, you can call them exploration and exploitation. So exploitation, kind of what they previously did, and exploration, they're kind of going in areas that they hadn't seen before. And you could actually tell which bird triggered the bout of exploration through this process. And we kind of would expect it always would be the naive bird, like the naive bird's kind of tugging the one with it. That's kind of like the narrative we would tell. But the weird thing about it is a lot of the bouts of exploration were triggered by the experienced bird, only in the presence of the naive bird. So there was like a team dynamic that was emerged from just having a new individual with the old bird that allowed the old bird to do something that hadn't done before.
I think that's the scientific manifestation of the feeling where if you listen to a song or you watch a video or you do something by yourself,
something you're working on,
or something that you're working on.
Sorry,
even, right?
We work on songs and videos. So.
Well, I'm even...
Yeah, something creative, and then you bring someone into the room to experience it with you. You see all these new things jump out.
And it's literally depending on who it is and how you know them and your relationship to them, the things that you will see are completely different.
Mm-hmm. Right. And yeah, and great, and that very well could be a great analogy for that.
That's
and
really
thank you for that. But the reason I brought it up is that, like, in theory, if you had a giant data set of people's actions over huge amounts of time, we might be able to start understanding the weird, like you might always expect everything's provost driven at a university. Like the provost makes a decision and that changes everything. But you don't realize as you bring new faculty in, they maybe have a plastic deformation on the provost herself. Or something like that. And in theory, maybe in the future, we could start to analyzing these things in this particular way and forming kind of new models of it. And the last kind of a quick example of that from the animal systems that I really like about this, and this isn't from my lab, but sort of this famous work in social foraging theory by, among other people, the kind of big research associated with it is kind of Luca and Giraldo, and where he would take birds, these little, I think they were spice finches, and he would put them on the ground and with little pods that maybe had seeds in them or maybe not. And so the birds would go around and you could watch their heads, and they would either be looking up or they'd be looking straight down, very clear. And the ones that were looking up, whenever another bird found seeds, it would run over and they would steal the seeds. They would exploit, that way. And so we called them scroungers. And the ones looking down, they wouldn't ever run to get the others while they're looking down, but they would find seeds and exploit the seeds they found to call them producers. Now, you put 10 birds together, and consistently, you'll always get certain birds being producers, certain birds being scroungers. And you put them away in cages, away from each other forever. You bring them back 8 months later, put them in the same thing, same 4 birds producers, same 6 birds scroungers. Now, you'd swap one bird out, doesn't matter if producer or scrounger, one bird with a novel bird, the mix totally changes. And that sounds pretty cool. But what was really cool about it is after Giraldo did that, then he took models from older optimal foraging theories, this thing called the marginal value theorem, and said if we run this little marginal value theorem program on modeled birds, we get the exact same pattern out. And so even though it had this really beautiful macroscopic pattern, it ends up being explainable by relatively simple kind of low-level rules. And so,
what is the marginal value theorem? What does it say?
The marginal value theorem basically says you continue to do a strategy until the instantaneous return from that strategy falls below the long-term return from the environment. So it's easiest to explain in terms of how long I stay in an area. So if I go to a bush looking for berries, if there are a bunch of bushes around so that it's really easy to find other bushes with other berries, I probably don't stay in that bush very long because the rate of return from the environment is really high. So my instantaneous return on that bush is going to quickly plummet below the long-term return. But if you start spreading those bushes apart, then I'm probably going to stay in each bush for longer because the rate of return from the environment is so low that I want to exploit one bush for longer. And you can generalize this idea to discrete strategies like producers and scroungers. And you can then predict if one bird is slightly better at something and another bird is slightly better at something else, then you can get them tending to be kind of producers for longer than other birds. And so you get this kind of diversity there. And when you put all the same birds together, you lock into the same pattern, but just a change of one bird, it's like a domino that changes all the rest of these. And so I bring this up because it shows that, going full circle to your original question about metabolism, there are simple rules of interaction that if we know the right rules, we maybe can find those interventions. We can find that one faculty member that kick out of the university and put the new one back in, and may change the entire system. But it's going to be subtle, and it's going to take a lot of thinking. And I think it's going to take not only mathematical models and computational models, but I think it's going to sort of accept that the world around us, there's organismal models of all sorts of beautiful social behaviors that we can borrow from when thinking about how we restructure our own human societies. So even though spice finches and ants and whatever else don't look like humans, they all represent to me different contexts that are thought-provoking. And that's why I think we should be stressing an emphasis on natural history and diversity, because I really think that diverse perspectives from nature help us enhance our diverse perspectives about our own systems.
It's beautiful.
I love it.
Yeah.
That seems like a good place to wrap for today.
Yeah.
I don't know if we,
I don't know if we can go up from there. That was very well said.
Thank you so much. Go ahead.
Oh yeah, where can folks find out more about what you're doing? Do you have a labs website coming up, or what's you do social media?
It's probably social media and my Google Scholar page. So the academics go to my Google Scholar. For pretty much anybody else, just find me at Twitter. I'm at Ted Pavlic, TEDPAVLIC, and feel free to reach out. always happy to talk about this stuff.
Excellent.
Thank you so much for joining us, Dr. Pavlic.
Yeah, thank you.
And don't forget to support us on Patreon, folks. We'll see you next week. Bye.
Bye, guys.